Good afternoon, everyone. My name is Akihide Kasai from Hokkaido University. Thank you. Okay, so my talk is about the offshore eDNA monitoring enabled by merchant ships. And in this initiative, scientists, the public, and private companies all work together to study biodiversity. Yes, as you can see in the figure on the left, this map shows the number of species in each region based on a well-known study by Costello et al. 2010. And every region has its own biodiversity, and it's encouraging to us to see that the waters around Japan rank very high. But today, I'm not focusing on where biodiversity is high or low, but instead I'd like you to notice the offshore areas shown in gray. These areas have little to no biodiversity data, mainly because biological surveys in the open ocean are very difficult to carry out. So we use environmental DNA, eDNA, to study biodiversity. eDNA makes field work much simpler than traditional methods, as has been discussed throughout this symposium. That said, we still need seawater samples. To study biodiversity in the open ocean using eDNA, we have to go offshore and collect water, which usually requires research vessels or training vessels from the university or some institute. And as a result, offshore surveys cannot be carried out very often. To overcome this limitation, we work with merchant vessels that routinely travel across the open ocean. For example, the Nippon Yusen Group operates a global shipping network, as shown in the figure on the left. And these maps show eDNA sampling carried out in the open ocean in 2025, in the last year. Yellow points and yellow lines represent sampling by scientists. And red points and red lines represent sampling by merchant ships. Research vessels, such as the Offshore from my university and Mirai from Jamstec, collected samples at many locations along their cruise tracks. The Ibaragi Fisheries Experiment Station also collected samples at a fixed point of the Joban areas of Tokyo. And on the other hand, merchant vessels collected samples on a regular basis. For example, Kinkai Yusen sampled once a month of Sandik area, and the Ogasawara Maru sampled once a month near Chichijima Island, as well as along the Tokyo-Chichijima route in August. In addition, Nippon Yusen company collected samples across the North Pacific using car and truck carriers. Since these ships do not follow fixed routes, sampling locations vary depending currents or winds. And to do that, before sampling began, we provided the ship crews with a detailed manual explaining the procedures. And the crew collected seawater from the engine cooling system or fire hydrants. And at the same time, measured water temperature and salinity. The data and the locations were recorded in a field notebook. And since filtration needs very careful handling, we tried to keep the sampling process as simple as possible and reduce the workload for the crew. So we developed a semi-automated filtering systems, filtering machines. It's equipped with 4 filtering cartridges, which is called Sterivex, and it can filter about 20 liters of seawaters in an hour. And the crew just collected 20 liters of seawater in a tank and put this hose, so this hose, into it. Once they hit the switch, the machine automatically pumps the water from the tank and runs the filtration. And when it's done, after the filtration, this device injects our DNA preservation solution into the cartridges on its own. Since the crew no longer has to handle the cartridge directly, and the whole process is much less stressful for them, I believe. And they put these machines itself into the fridge and keep it in the cool conditions. And after getting all the ships, they send that machine to us, and we are going to analyze eDNA from these samples. And now this device is being commercialized by a startup company that originates at Jamstack. So this is examples of eDNA results from merchant ships in spring 2025. At each site, the top 8 detected species are shown in this figure. As far as we know, there have been very few cases where marine fish communities were surveyed at so many widely spread points within such a short time. And we found that each sampling site has its own distinctive set of species, fish species. For example, Pacific Soli, Samma, is very important, which is one of the very important fishery resources for Asian people, was detected at stations, which one? 1 and 6 or something like that. 3 and 6, yes. And Japanese sardine was also very important fishes, was found at the western stations of the Pacific, while California sardines was found at the eastern stations in the Pacific, right? These patterns are consistent with previous works, previous studies, demonstrating that eDNA sampling using commercial vessels can reliably capture fish community structures. Okay, this is another example. Near Ogasawara Islands, 85 species were detected in 5 months monitoring. Among them, 31 species were coastal tropic fishes. Yeah, like these ones, very cute, right? And in that site shows a very clear seasonal pattern, even though it is located in the subtropical region. The water temperature still changes with the seasons, low in winter and very high in summer. It's about 23 degrees Celsius in winter and increased about 30 degrees in summer. We also see the fish community shifting with these temperature changes. Diversity is low in winter, but it becomes very high in summer. Next, I will show you some examples that was conducted by research vessel Mirai from JAMSTEC. Seawater was pumped up at multiple stations along our voyage from Japan to Arctic. And we filtered the seawater to examine changes in fish communities along this line. In this survey, a total of 107 species were detected. And the figure on the left shows fish species on the vertical axis, and the sampling sites on the horizontal axis, with detected species highlighted in blue. And darker colors indicate higher concentrations of eDNA. For example, flying fish were detected at southern sites, whereas Arctic cod were detected only in the Arctic Ocean. And the above of the heat map on the left-hand side is a dendrogram representing the results of the cluster analysis. The figure on the right shows species richness on the vertical axis and latitude on the of the sampling sites on the horizontal axis. Yeah, it is clear that the more fish species were detected at lower latitude. In particular, there is a distinct boundary around 40 degrees north. Yeah, more than 20 species were detected south of this latitude, whereas only about 10 species or fewer were detected to the north. And from an oceanographic perspective, this boundary represents to the transition zone between the Oyashio and the Kuroshio currents. I mean the subtropical and the subarctic gyres. And the other boundary is here, which is close to the Aleutian Islands. And detected fish species were different in the north and the south of the this front. On the other hand, the Bering Strait between the Bering Sea and Arctic Sea doesn't seem to form a strong boundary, indicating that the fish migrates through this Bering Strait. Yeah, as is shown in the Kawamiya-san's talk, the Kuroshio current has a strong effect on the marine environment and ecosystems around Japan. As you know, until a couple of years ago, the Kuroshio current exhibited a large meander south of Japan, and as a result, the Kuroshio Extension area, which normally flows eastward, shifted northward as far as off northeastern Japan. As a result, yeah, seawaters as far north as off Miyagi Prefecture remained about 4 degrees Celsius warmer than average for several years. And however, since last year, the northward shift of the Kuroshio extension has finally subsided, and conditions are gradually returning to normal. This change in Kuroshio path should have some effect on the fish structures of Tohoku area. Then rural ship Mashi Umaru have been sampling seawater of Tohoku for several years. So I analyzed changes in fish communities. The waters were sampled at these red points. I'm not listing all of the detected fish species here. Instead, I present examples of warmer water fish in red and cold water fish in blue in the list. And in 2023, in the left-hand side, in this year, the Kuroshio extension flowed further north. Japanese eel was detected of Sandic area. In 2024, black sea bream and tuna, both of them are very warm fish, were detected, but they are not in 2025. Okay. It is interesting that demersal fish, comparing with the pelagic ones, demersal fish communities did not change so much. Okay. And this is another research results from training ship Oshoromaru from Hokkaido University, from our universities. And the figure in the left shows the sampling points, and the middle shows the relative proportions of lead counts of each fish detected in each sample. And the figure in the right shows the temperature and salinity along the cruise line. Sampling points in the north were occupied by low temperature here, low temperature and low salinity of Oyashio water, while in the south, here, the Kuroshio water approaches coast as shown in high temperature and salinity. And here, Japanese anchovy, shown in green in the middle figure, was detected across the entire study area and was dominant at many stations. Mackerel in the yellow were mainly detected in regions where the Kuroshio approaches the coast, while rarely detected at more northern stations. The orange-colored flounder, halibut, detected at several key locations between 37 and 38 degrees north. This is a coastal species because the sampling sites in these areas were particularly close to the coast. And its detection reflects coastal community structures. And at last, the sardine shown in purple were proportionally more abundant, more dominant at the northern locations. And based on the break cassis distance, the sampling sites were grouped into 4 clusters. These clusters were primarily separated by latitude, north, south, and middle, but some sites were classified into a coastal community. In the MDS plot here, these ones, the clusters were also clearly distinguished from one another, and especially the northern group and the southern group. Northern group is here, and the southern group is here, are widely separated on this plot, and indicating that their fish species compositions are differed greatly. And these contours, blue contours, represent isotherms. The northern and the southern regions were separated in accordance with temperature gradient, and while the coastal regions, here, this one, formed distinct clusters driven by factors other than temperature, that is, salinity. In addition to the last year's sampling, we plan to collect water samples using 2 more vessels this year. One is Wild Rose Leader, a car carrier cargo, and the other is Le Soleil, French one, a luxury cruise ship. By expanding our sampling network in this way, we hope to capture an even broader pictures of marine biodiversity across the open ocean. Ultimately, our goal is to build a large-scale biodiversity dataset within the support of merchant vessels operating around the world, and to contribute to a better understanding of how ocean ecosystems are changing. Thank you very much for your attention.